All right, everybody. Welcome. My name is Anna Stracolini and my partner is Kendall Hart, and today we're going to be talking about the phospholipid bilayer, phospholipids, and overall the structure of the cell membrane. So the first thing I'm going to be talking about is a phospholipid. These are basically composed of two parts, a head, which is hydrophilic, meaning water-loving, and a tail, which is hydrophobic, meaning water-fearing. These are water-loving and water-fearing because of their charges. And overall, a phospholipid is amphipathic, which basically just means that it is composed of two different parts with two different charges. And since the head is hydrophilic and the tail is hydrophobic, a phospholipid is consisting of two separate parts. Alright, so the next thing I'm going to be talking about is the phospholipid bilayer. So bi means two, which basically means that the phospholipid bilayer is two layers of phospholipids. However, these phospholipids are facing in opposite directions so that the hydrophilic heads are on the outside and the hydrophobic tails are on the inside. So when the phospholipid bilayer is in a watery environment, the water-loving heads will be on the outside surrounded by water, but the water-fearing tails will be on the inside where they will not be surrounded by water. So the next thing I'm going to be talking about is integral proteins. And these are amphipathic, which, as we learned earlier, are basically a molecule that has two different types of affinities, which is really a polar end that is attracted to water and a nonpolar end that is repelled by it. So these integral proteins are in the plasma membrane and they pass throughout the entire membrane. And they are also transmembrane proteins since they pass throughout the entire membrane and they are permanently attached. The next thing I'm talking about is peripheral proteins. And peripheral proteins are both polar and hydrophilic, which means they are attracted to the water and they are also water-loving. So they will be on the outside of the plasma membrane where they will be surrounded by a watery environment, which they are totally fine with, and they are attracted to that type of environment.
This is cholesterol. Cholesterol is a steroid that is found only in animal cells, and its main function in the membrane is to maintain the structure. These are glycoproteins. Glycoproteins are phospholipids and carbs in one, and they maintain the structure of the membrane and also help cells differentiate between self and non-self cells, basically meaning that self cells are just body cells, while non-self cells are foreign cells or cells that do not originate from your own body. These are channels. Channels allow for passive transport, and passive transport means that you do not need energy to transport. Their main function is to get things in and out of the cell. Carriers are proteins. These proteins bind to one side of the membrane and then change their shape to be able to transport a material across the cell. Recognition helps certain proteins to help the cell in differentiating between self and non-self cells, which is very important in triggering an immune response. And again, self cells are just body cells, while non-self cells are foreign cells that do not originate from your body. Receptors are proteins that usually span the whole membrane to relay information from inside and outside the cell in order to communicate throughout the membrane. Enzymes are also proteins that enhance the rate of reactions that happen at the membrane level by lowering the activation energy needed to activate the enzyme.